Friends, I am Dr. K.D. Mishra and today we will discuss the process of hemopoiesis. Hemopoiesis means formation of blood, especially formation of the structural elements of blood, which includes RBC, WBC and platelets. This is also called as hematopoiesis. Hemopoiesis or hematopoiesis, the meaning of this word, when we say hemo means blood and poiesis means production. There is a system of organs which is responsible for production of blood cells. It is called as hemopoietic system. You know, in different stages of life, different organs functions as hemopoietic organs, which include bone marrow, which is the main part where hemopoiesis occurs. Then lymph nodes, again here the formation of lymphocytes occur throughout the life. Then spleen and liver, in spleen and liver, during the fetal life, that is intrauterine life, the blood cells are firstly formed into the yolk sac, and then in the second trimester the blood cells are formed in spleen and liver. Then comes thymus. It is one of the important organ where not only the lymphocytes are produced, but the lymphocytes matures into T lymphocyte to perform specific functions in immunity. Now, blood cells, that is RBC, WBC and platelets, have a definite lifespan and they are regularly destroyed and have to be replaced. The process of formation of blood cells is called hemopoiesis or hematopoiesis. Now you see, the bone marrow produces as much as 6 billion blood cells per kg body weight per day. Such a huge amount of blood cells are to be produced to maintain the count of blood cells in the blood. This includes production of 2.5 billion RBCs, a similar number of platelets and about 1 million WBCs. Now, the site of hemopoiesis depends upon the stage of life. That means in embryo, that is intrauterine life, the hemopoiesis occurs mainly in yolk sac. But later on, in the intrauterine life itself, the hemopoiesis shift to liver and spleen. And after 5th month or so, that is during the nearly about 5 months onward, the hemopoiesis is confined to bone marrow. We call it medullary hemopoiesis. Now, the phases of hemopoiesis, if we divide these, one is the embryonic or prenatal phase. That includes mesoblastic phase, hepatic period and myeloid period. Mesoblastic period is the formation of blood cells in the yolk sac. Hepatic period, that is the formation of blood cells in liver and spleen. And myeloid period, that is the formation of blood cells in the bone marrow. Now, in postnatal life, that is after birth, the hemopoiesis is confined mainly to the bone marrow. It is called medullary hemopoiesis, whereas in certain cases when the number of blood cells cannot be maintained by production into the bone marrow, then it is sometimes taken up by liver and spleen. That is called extramedullary hemopoiesis in case of adults. Now here in this picture you can see the hemopoietic organ, especially during the intrauterine life. So first phase is the mesoblastic stage. This is during the first trimester. The yolk sac contains cells which are multi-lineage differentiating capacities. That means they can form different types of blood cells. Such cells are called multi-lineage cells. Then in the second trimester of fetal life, the hemopoiesis, that is formation of blood, shifts to liver and spleen up to 5th week, from the 5th week to 5th month. Then comes the final stage, that is medullary stage, in which in 5th month of fetal life, bone marrow becomes hemopoietic, and here erythropoiesis begins towards the end of 3rd trimester. So first of all, the hemopoiesis occurs in the bone marrow, that is the production of other cells than RBC, but RBC production only starts at the end of 3rd trimester. Now we'll take the stages one by one. If you see in mesoblastic stage, during embryonic or intrauterine life, yolk sac contains cells with multi-lineage differentiating capacity. That means they can differentiate into different types of blood cells. Some non-yolk sac regions are also included in production of blood cells. One of these is PSP, that is para-aortic splanchnoplure, which give rise B series, and another is AGM, that is aorta gonad mesonephros, which give rise pluripotent cells. Now the lymphohematopoietic cells, which produces lymphocytes, have been detected in the 9th day of yolk sac. That means the stem cells which forms lymphocytes are formed as early as the 9th day of yolk sac formation. Now the second stage during intrauterine life of hemopoiesis is the hepatic stage. During 2nd trimester of pregnancy, hemopoiesis starts in liver and spleen. It is called hepatic stage. In liver, hemopoiesis occurs, it starts as early as 5th week and reaches its peak in the 5th month of gestation period, and finally ceases at birth when it is totally taken over, that is the production of blood cells is totally taken over by the bone marrow cells. Now the medullary stage. Hemopoiesis in bone marrow is called medullary hemopoiesis. In 5th month of fetal life, bone marrow becomes hemopoietic. Initially, only granulocytes and megakaryocytes are produced. That means initially in the medullary stage, the bone marrow forms granular leukocytes and the megakaryocytes. The granular leukocytes include the eosinophil, basophil and neutrophils, whereas the megakaryocytes are responsible for production of platelets. In bone marrow, erythropoiesis begins towards the end of third trimester. That means before that it is formed in the liver, but after that in the third trimester the erythropoiesis starts in bone marrow. Now coming on to medullary erythropoiesis. After birth or during the postnatal life, the process of erythropoiesis, that is the formation of blood cells, totally shifts from other hemopoietic organs like yolk sac or liver, spleen to the bone marrow. But in certain conditions, when the blood cells are not sufficiently produced from the bone marrow, liver and spleen take up the production of blood cells, that is, they take up the hemopoiesis in emergencies. This is called non-physiological hemopoiesis and it is considered an abnormal phenomena. Now, sites of medullary hemopoiesis. As I mentioned just now, that medullary hemopoiesis means hemopoiesis in bone marrow. It occurs in the red bone marrow of long bones. As you can see that in long bone marrow, in this portion, it has cancellous bones where blood cells are formed. So, there in the long bone marrow, long bones, red bone marrow is present where hemopoiesis occurs. Then erythropoiesis occurs continuously in bone marrow of vertebrae, ribs, sternum, skull, pectoral and pelvic girdles. Erythropoiesis regresses. That means the speed of production of blood cells, it is reduced or regresses after 10 years of age. Now coming on to the steps of hemopoiesis, bone marrow contains two types of cells. One is hemopoietic stem cells, which are responsible for production of series of cells which produces the final leukocytes or RBCs or platelets. And second type of cells which are found in bone marrow are the cells which are produced by hemopoietic stem cells. They are called as progenitor cells. If they are responsible for producing erythrocytes, they are called erythrocyte progenitor series. If they are responsible for the production of leukocytes, they are called leukocyte progenitor cells. If they are responsible for production of platelets, they are called megakaryocyte progenitor cells. The stem cells develop into progenitor cells which ultimately differentiate into different types of blood cells. That is what I said just now. Here you can see the steps in hemopoiesis. The pluripotent cells, that is PPSC, are the mother cells. They are also called hemocytoblast. A blood blast, when used before a particular cell, that means it is the mother cell or stem cell which produces that particular type of cell. So, the PPSC are multilineage stem cells that produces two groups of stem cells. One group is that of myeloid stem cells. It is multilineage stem cells which produces many types of blood cells, whereas another type is lymphoid stem cells, which is unipotent and produces only lymphocytes. Here you can see in this picture, this is the pluripotent cell, which is a stem cell. It produces two series. One is myeloid stem cell, which produces different types of blood cells, whereas the other one is lymphoid stem cell. It is unipotent and can produce only lymphocytes. Now, myeloid-erythroid ratio. As you know, the blood cells are continuously destroyed and replaced. In bone marrow, cells of myeloid series are 70 to 75%. In comparison to erythroid series, the erythroid series is responsible for production of erythrocytes or RBCs. The number of RBC and WBC in the circulating fluid is in the ratio of about 1:700. That means one white blood cell WBC to 700 RBCs. But here if you see the difference in these percentage, if I write WBC, that is 75%, and RBC, that is 25%. Okay. Now this ratio come 3:1. That means the production of myeloid series, which produces WBC, is more, 3 times more as compared to the production of erythrocyte series, which produces RBC. It is almost 3:1 ratio. But when we see these cells in circulating fluid, then the ratio comes 1:700. That is, for one WBC, there are 700 RBCs. Why this difference is there? Why the cells producing WBC are more, but number of WBC is less in circulating blood? This is because of the lifespan of different types of blood cells. As you know, the lifespan of RBC is 4 months, that is 120 days, whereas the lifespan of WBC is just few hours to few weeks. Therefore, the production of RBC should be maintained at a higher rate so as to maintain a balance between the proportion of these two types of blood cells. The myeloid stem cells, as I mentioned earlier, these are pluripotent cells. Those are capable of forming 3 different progenitor cells. Progenitor cells are those cells which give rise to the final cells. Number 1 is erythroid progenitors, which produces erythroid series, which ultimately produces RBC. Another series is granulocyte progenitors, which produce granular leukocytes, which finally produces neutrophils, basophils, and eosinophils. The third type of progenitors are the monocytic progenitors, which produces monocytes, and then the lymphocytic progenitors, which forms lymphocytes and megakaryocytes, which ultimately form platelets. Now, the lymphocyte stem cells, as I mentioned just now, they are unipotent stem cells. We call it UPSC, and these are capable of producing only one type of cells, that is only lymphocytes. Later on, these lymphocytes are differentiated into B lymphocytes and T lymphocytes. You know, here in this picture you can see the lymphocyte progenitor produces B lymphocyte and T lymphocyte. B lymphocyte are called bursa equivalent lymphocyte, which are produced in bone marrow. Bursa is a part, in case of bird, it is situated near the cloaca. So these cells are equivalent to those cells, therefore they are termed as B lymphocytes. Then T lymphocytes are those which are produced in bone marrow, but they get matured in the thymus, a T for thymus, so therefore they are called T lymphocytes. Now here in this picture, you can see the whole process of formation of blood, that is hemopoiesis. If you see here, this is the pluripotent hemopoietic cell, which produces other PHSC, that means it produces many pluripotent hemopoietic cells, which then differentiate into 2. One is colony forming unit, spleen, which will differentiate into erythroid series and granulocyte series, and one differentiate into lymphocyte stem cells. These lymphocyte stem cells then differentiate or develops into B lymphocytes and T lymphocytes. On the other hand, the colony forming units are formed from the pluripotent hemopoietic cells, which forms 2 series. One series is colony forming unit erythrocyte, which ultimately produces RBCs, and another is colony forming unit GM. Here G is for granulocyte and M is for monocytes. So this form 2. One is granulocyte series, which further differentiate into neutrophil, eosinophil and basophils. Another is monocyte series, which produces monocyte, which are phagocytes. Now another cell differentiated from this is colony forming unit megakaryocyte. These megakaryocyte develops into this CFU megakaryocyte develops into megakaryocyte, and megakaryocyte finally develops into platelets. So this is how a pluripotent hemopoietic stem cell differentiates into different series to give rise lymphoid stem cell, then colony forming unit, granulocyte and monocyte series, then erythrocyte colony forming unit, and the lymphocyte further differentiate into T lymphocytes and B lymphocytes. Now, this is a different representation of hemopoiesis. Now, here you can see the hemopoietic mother cell here, which differentiate into different types of different series, which finally develop into blood cells. Here you can see the erythrocytic series developing into red blood cells. Here you can see the megakaryocyte series, which develops into platelets. Here you can see the monocytic series, which develops into monocyte, and then these monocytes are shifted to the tissue, forming tissue macrophages. Then there is granulocyte series, which finally produce neutrophils, basophils, and eosinophils, and the lymphocyte series, which finally produces B lymphocytes and T lymphocytes. Yet another presentation of hemopoiesis, which I think is more simpler than the previous two. Here you can see the hemocytoblast cells, that is pluripotent stem cells, PPSC, which differentiates into 1, 2, 3, 4, 5. One is proerythroblast series, which finally develops into erythrocytes. Another is myeloblast series, this one, which develops into 3 neutrophilic myelocytes, then eosinophilic myelocytes and basophilic myelocytes, which finally develops into neutrophil, eosinophil and basophil. Here you can see the monocytic series, which develops into promonocyte and finally into monocytes. The lymphocyte series, which finally develops into lymphocyte, and these lymphocytes, they finally develop into B lymphocyte and T lymphocyte. And here is megakaryoblast cell, which developed into megakaryocyte, and megakaryocyte finally fragments into the platelets. That is how all the components of blood are formed. Now, regulation of hemopoiesis. Hemopoiesis is regulated by cytokines, commonly called hemopoietic growth factor, HGF. One of the important hemopoietic growth factor is erythropoietin. Erythropoietin is secreted in the kidney, in hemopoietic tissue. You know, between the kidney tubules, there is an interstitial cell which is called hemopoietic tissue that secretes erythropoietin, and this proteinaceous hormone stimulates formation of precursor of erythroid series. Now, second one is interleukins, IL. It controls development of eosinophils, lymphocytes and monocytes. Third one, colony forming stimulating factors, that is called CSF. CSF are grouped into 3 groups. Now here M is for monocyte, G is for granulocyte, GM for granulocyte and monocyte, and G for granulocyte only. So these are named as MCSF, GCSF, that is monocyte colony stimulating factor, granulocyte colony stimulating factor, GM, granulocyte monocyte colony stimulating factor, like that. Now here is the importance of production of blood. especially RBCs. If you can see in this picture how much RBCs are destroyed, about 2 million RBCs are destroyed and replaced every second. You can just imagine what a huge number of RBCs are destroyed every second. And they, for maintaining the number of RBCs, that means for maintaining the functioning of RBC, that is mainly the supply of oxygen to the tissue and cells, the number of RBC has to be maintained at 5 to 6 million per cubic millimeter. Now here you can see in circulation, the RBC number is 10 to the power 3 into 10 to the power 13, which amounts to 900 gram of total hemoglobin. Out of this, about 1 into 10 to the power 10 RBC are destroyed per hour. That means 0.3 gram of hemoglobin is destroyed per hour. Now, this goes to the spleen, where blood cells are destroyed, then these are converted into bile pigment, and bile pigment passes from the stool and so on. That we will discuss later on. But due to these, the iron is released, that iron is back to the liver for formation of hemoglobin. The proteins are broken into amino acids, and those amino acids are again used for synthesis of proteins. Then 1 is to 10 to the power 10 RBC, that is 0.3 gram of hemoglobin per hour is produced to maintain the number of RBC and the amount of hemoglobin in blood. Now coming on to the erythropoiesis. Erythropoiesis, that means formation of RBCs. The progenitor cells give rise colony forming unit erythrocyte. That means CFUE, colony forming units that are responsible for formation of series of cells which form finally erythrocytes. Now erythropoietin is the hormone which stimulates differentiation of erythroid series, lineage, which forms different stages in erythropoiesis. That means erythropoiesis is the main hormone which is responsible to stimulate the production of RBCs. Now, stages of erythropoiesis. You know, hemocytoblast cells, those are the mother cells. These are large cells of 18 to 23 μm diameter. They have large nucleus and basophilic cytoplasm. These cells are divided to produce proerythroblast cells, which are 14 to 19 μm in diameter. Their cytoplasm is basophilic. They have large nucleus and there is no hemoglobin in these cells. Then they are stimulated to produce early normoblasts, which are still smaller, 11 to 17 μm in diameter. In these, the nucleoli disappears and they actively starts dividing. The next stage is the intermediate normoblast tail. It is very important stage in the process of erythropoiesis. As the cells becomes still smaller, that is about 10 to 14 μm in diameter, the nucleolus disintegrates, nucleus condenses, that means the nuclear material condenses further. Now here the important thing is that the formation of hemoglobin starts here. This is the stage where hemoglobin formation starts and cytoplasm becomes colorful, that is, it becomes polychromatic. Now, this stage, that is intermediate normoblast stage, do not divide further, it just matures. It do not divide and it forms normoblast. Now, in the late normoblast stage, the size of the cell is further reduced to 7 to 10 μm. Nucleus becomes very small and dense. It is deeply stained and the amount of hemoglobin increases because from the previous stage, synthesis of hemoglobin has already started. It further matures and lead to complete loss of nucleus. So, the nucleus is completely lost. As you know, the RBCs are non-nucleated cells, and now it passes to the next stage, that is reticulocyte stage. In these, the cells have reticulum in the cytoplasm, therefore they are called reticulocytes. They are stained with vital stains like chrysal blue. The reticulocytes finally mature into erythrocytes. From proerythroblast, that is megaloblast, to reticulocyte stage, it takes about 7 days. And from reticulocyte, that is from here, to the maturation of erythrocyte, it takes another 2 days. That means formation of erythrocyte from proerythroblast stage to erythrocytes, that is mature RBC, takes about 9 days. Now here in this picture, you can see the complete process of erythropoiesis. This is the proerythroblast cell with large nucleus, then basophil erythroblast. This is called early normoblast stage, where you can see the size of the cell is reduced, but the nucleus remains there, it is larger. Then the polychromatophil erythrocyte, that is intermediate normoblast stage. Again here the size reduces. Then the late normoblast stage. This is the important stage in the process of erythropoiesis, where the formation of hemoglobin starts. And then in the next stage, which is called as reticulocyte, in this stage the nucleus is finally disintegrated and the erythrocytes are matured. Now, erythrocyte stimulating factor, that is ESF. There is a glycoprotein hormone produced in the kidney, mainly in kidney. It is produced 90% in kidney, and some amount of it is produced in the liver. As soon as a decrease in oxygen is felt in the blood, that means a condition of hypoxia is seen. It may be due to many reasons like anemia, it can be due to low blood pressure, it can be due to low hemoglobin concentration, it can be due to low blood supply, it can be due to any kind of disease concerning to lungs, that is pulmonary diseases, or it can be at high altitudes. In all these conditions, the amount of oxygen in the blood, which is carried out by the hemoglobin, is reduced, and this results in tissue hypoxia. When such a blood reaches kidney, the hemopoietic tissue of kidney releases a proteinaceous hormone, that is erythropoietin, and this erythropoietin stimulates bone marrow to secrete, produce more number of erythroblasts. In such circumstances, erythropoietin secretion increases, which in turn enhances RBC production until this condition of hypoxia, that is deficiency of oxygen, is relieved. Now here in this picture you can see how this erythropoietin hormone works. Now in any one of these conditions, maybe due to low blood volume, maybe due to anemia of any kind, maybe due to reduction in the number of RBCs, maybe due to decrease in the hemoglobin percentage, maybe due to any abnormal type of RBCs like sickle cell anemia, macrocytic anemia, or megaloblastic anemia. In any of that case, or in case of poor blood supply, if the blood supply is poor, it's less, if the heart is not working properly and blood is not supplied properly to different organs, or any kind of lung diseases, that is pulmonary diseases, when lungs are not properly working, the oxygen is not properly diffused from the lungs into the blood. So the oxygen supplied to the kidney, if it is deficient in oxygen, then this hormone hemopoietin is secreted. Now this hemopoietin, when it reaches to the bone marrow, then it stimulates the hemopoietic stem cells, which produces proerythroblasts, and it goes through the series of formation of erythroblast series, and finally production of red blood cells is increased. That is how the production of RBCs is maintained in the body. Now maturation of RBC, as we have seen that after late normoblast stage, they do not divide further, the RBC matures. Now for maturation of RBC, DNA is required, or for that there are two vitamins which are necessary for maturation of RBCs and for production of DNA, that is the vitamin B12 and folic acid. They are important because these are needed for synthesis of DNA. Specifically, these two vitamins are required for the conversion of proerythroblast to early normoblast stage or erythroblast stage. Deficiency of these, now say, what happens in the final stages of RBC maturation? That is, the nucleus gets condensed, it becomes smaller and smaller, and then finally the nucleus disappears. This happens to accommodate more amount of hemoglobin in the RBC, so that more amount of oxygen it can transport from the lungs to the tissue. Now deficiency of these vitamins, when deficiency is there, the RBC do not mature, they remain larger in size with nucleus. So this is called as macrocytic anemia or megaloblastic anemia. Another term is megaloblastic. That means the RBC do not mature, they remain larger in size. So this is a kind of anemia because although blood cells are there, but the amount of hemoglobin is less, they are nucleated, their shape is larger. So when they pass through the spleen to the tortuous network of capillaries in the spleen, they are broken and anemic conditions are produced. So therefore it is called macrocytic anemia or megaloblastic anemia. Now maturation of RBC, again, some metals are required for that. Iron is the basic metal required for synthesis of hemoglobin, along with some amount of copper, then manganese, cobalt, calcium. These are important for synthesis of hemoglobin. Now, for absorption of these metals from the food in the alimentary canal needs an stimulation by the bile salts. Bile salts stimulates the absorption of these metals. So, if there is a deficiency of that bile salts, then absorption of these metals will not take place, or if there is a deficiency of these metals itself, then also production of RBC will be affected. Now vitamins plays a very important role in the maturation of RBC. Vitamin C, vitamin B6, vitamin B12 and folic acid we have just discussed, riboflavin, pantothenic acid, nicotinic acid are important for RBC maturation. Now, as just mentioned that vitamin B12 is essential for maturation of RBC because it is responsible for synthesis of DNA. But you know, even if vitamin B12 is sufficient in the diet, and if intrinsic factor is deficient, that means if intrinsic factor is not there, you know, intrinsic factor is secreted from the stomach by the parietal cells of gastric gland. This intrinsic factor is responsible for absorption of vitamin B12 from the intestine, so that if deficiency of intrinsic factor is there, a type of anemia occurs that is called pernicious anemia. It is caused due to loss of RBCs. That's all for today. Thank you very much.